Okay, now, we focus on dihybrid inheritance. Okay, from the word dihybrid, okay, di means double, okay, two. Okay, so what is dihybrid cross? Okay, dihybrid inheritance involve inheritance of two characteristic. Okay, so we need to cross two characteristic at the same time. So each characteristic is controlled by a different gene. Means there are two pairs of alleles, okay, located at different locus. So each alleles to girls, pairing of alleles to, they're located at the different locus. Example, okay, in monohybrid inheritance, okay, kita kacuk one characteristic only, right? Okay, this is homologous chromosome. Okay, homolog dia mesti same size. Walaupun I lukis macam tak berapa sama kan, tapi sepatutnya dia same size. Okay, so the gene must be located at the same locus. Okay, so this is one trait. Okay, ataupun kita panggil a pairs of allele. So dia akan show the characteristic, okay, of the organism. So ini baru satu ciri. Okay, then dia ada lagi one more characteristic, okay, from the pairs of allele, different pairs. So dia datang daripada different location. So that's why for today, we are going to do the schematic diagram for dihybrid. Okay, maksudnya untuk dua ciri, okay, dua karakteristik at the same time. So dia punya gamet akan jadi lebih banyak, ya. Okay. So, schematic diagram of a dihybrid inheritance between two purebred parents to study the characteristic of P, namely seed color and seed shape. Okay, so for this case, hari ni kita cuba kacukkan, okay, kita nak cross between the characteristic seed color and seed shape. Nampak tak kita ada dua karakteristik? Okay, kalau in our previous class, only one trait, okay, about the height ataupun about the color of fur. But now there are two traits. Ada dua traits yang menunjukkan two karakteristik lah. Okay. So, the two purebred parent either have a pair of homozygote dominant allele or a pair of homozygote recessive allele. So, apa yang kita akan buat ni? Kita akan cross between pair of homozygote dominant, okay, cross with the pair of homozygote recessive. Kita akan kawinkan homozygote dominant dengan homozygote recessive. Okay. So, fertilization between the two purebred parent produce offspring of F1 generation with the dominant trait of round and yellow seed. So, kita akan nampak nanti the F1 generation, okay, filial 1, generation yang pertama, all are dominant traits, which is round and yellow seed. Okay, so kita tengok dia punya picture. This is what Mendel do for Mendel second law, Mendel second law. Okay, so this is the key. Key ni biasanya dalam exam dia akan bagi. So we need to write. Bila kita tulis, so kita tahu, okay, what is the genotype, okay, that we need to write in the schematic diagram. Okay, so big B is dominant allele for round. Okay, so small b must be recessive lah, okay, for constricted. Okay, big K dominant for yellow, small k for green, which is recessive. Okay, kalau you tengok first parent phenotype dia ni, okay, round and yellow seed. So kita tengok dekat sini, round and yellow both are dominant. So we need to write the genotype, okay, parental genotype, big B, big K. Because first yang Mendel buat, dia mesti kacukkan purebred. Maksudnya both are dominant. Okay, homozygote dominant. Alright, so dia cross, tolong tulis cross ni, I tak tahu kenapa textbook ni dia lost dia punya cross, dia missing. Tolong tulis sebab penting ni. Okay, cross between the constricted and green seed. Okay, so this is recessive. Both are recessive. This is homozygote recessive. Okay, so look at the picture. Ada gambar kan, tapi dalam exam tak payah lukis gambar ni tau. You just write the phenotype and genotype. Nanti dalam you punya schematic diagram, dalam paper tu, you tak payah lukis ni. just write the phenotype and genotype only. Okay, so then the genotype for the recessive homozygote recessive ni, small b, small b, small k, small k. Alright, so this is called as the purebred parent. Okay, so then from the parent genotype, okay, we take out the meiosis. Okay, draw an arrow. Okay, arrow, arrow. Okay, take out gamete in circle. Okay, big K, big B, big K. Small b, small k. Then you fertilize it. Okay, tulis fertilization, fertilization, arrow. So then F1 genotype. So after parent must be F1 genotype. Yeah. Okay. Then big B, small b. How come you get this? Okay, from the gamete. Big B, small b. Okay, settle for the shape. And then big K, small k. This one for the color. Okay, so from the genotype, okay, we can determine the phenotype. Okay, so we follow the dominant, right? So big B must be round. Big K, dia mesti follow big K. Small k ni recessive, so they must follow the color for dominant, which is yellow. So all round and yellow seed. That's why tadi dalam previous slide dia kata for F1 generation, this is a dominant. Dia carry the dominant characteristic. Nampak tak? Dia round and yellow. Okay? So tapi dia tak settle lagi, baru dapat F1 generation. So dia pun buat lagi sekali, Mendel ni. Dia buat satu kali lagi fertilization between the F1 generation to get F2 generation. Samalah macam kita buat monohybrid. Okay. So kita pun take out the, kita tulis dulu phenotype dia. Okay, round and yellow seed. Okay, cross with round and yellow seed. Okay, so the genotype, F1 genotype. Okay, kita copy je lah Okay, big B, small b, big K, small k, cross. Big B, small b, big K, small k. Right? So, meiosis, we take out the gamete. There are a lot of variation. Banyak variation dia. Sebab tu lah I kata topik ni dia ada kaitan dengan probabilities, subject maths. So, apa yang kita buat ni? Kita buat dia punya probabilities. Kita andaikan berapa ratio, okay, untuk dia punya next generation. Because gamete ni ada banyak variation. Okay, so from this genotype, we can get 4 type of gametes. Okay, so big B, big K. This is both dominant. Big K, big B, big K. And then there maybe ada possibility for big B, small k. Okay, then small b, big K. And lastly, both are recessive, small b, small k. Nampak? So for the other side, same also the gamete because same genotype. Okay, so for this case, is it possible for us to cross one by one? Possible tak kita nak cross one by one macam Mendel First Law? Okay, tak logik kan girls? So nanti kita akan jadi pening kepala. Okay, how come we can cross one by one? Because there are 4 and 4. Okay, so that's why for this we need to use Punnett Square. Okay, untuk Mendel dihybrid, dihybrid Mendel second law. Okay, I want you to draw the Punnett Square for F2 generation. Okay, when we cross the gamete. Okay, so nanti kalau untuk monohybrid, you tak payah lukis Punnett Square. You just cross je because dia maximum pun dia just ada 2 gamete, 2 gamete. That one tak ada masalah lah, you just cross. Okay, but for this, nampak during formation of gamete, any allele for seed shape can pair with any allele for seed color. So there are a lot of variation for gametes. Okay, that's why we need to draw the Punnett Square. So Punnett Square ni bentuk macam ni lah. Okay, so we need to arrange the gamete one by one. We put in the box. Okay, dia ada 2 arrows, X and Y axis lah. Okay, so big B, big K, kita letak satu table. Big B, small k, small b, big K, small b, small k. Susun lah. And then you put at the Y axis, sama je, susun sama. Big B, big K, big B, small k, small b, big K, small b, small k. Okay, then you need to cross it. Cara cross dia, you senang je, you settle one by one. Okay, first you cross yang ni dengan ni. So big B, big B, big K, big K. Put the genotype first. Settlekan genotype, susun tulis dia punya genotype dulu. And then bawah dia, you tahulah dia punya phenotype is what? Round and yellow lah, because both are dominant. Ya, big B and big K. Okay, then you cross yang ni dengan ni pula. So you dapatlah big B, big K, small k. So what is the phenotype? Round and yellow, because they follow the dominant K. Okay, this one, big B, small b, big K, big K. Okay, yang ni dia settlekan this row dengan yang ni, kolom yang ni. Ya, okay, big B, small b, big K, big K. So the color, the shape must be round and yellow. Okay, then last one, for this row, okay, for this column and this row, okay, big B, small b, big K, small k. So the round shape and yellow color. So you settle lah one by one. I tahu you faham kan, you ambil row then you cross dia dengan dia punya row dengan column dia. So you can get the genotype and then you write the phenotype. Okay, so you tak payah lukis lah ni, tak payah lukis. Dalam exam pun tak payah lukis ni, dia nak tunjuk dekat you. Okay, so this is the result for Mendel second law ataupun kita panggil dihybrid inheritance. Alright, so daripada sini, we can come up with the ratio also. Okay, I nak tanya you, what is the ratio for monohybrid inheritance in our previous class? Monohybrid, what is the ratio?
3-1.
Okay, anyone please answer?
3-1, teacher.
Monohybrid, what is the ratio? At the last, you dah lukis dua schematic diagram hari tu kan? From textbook and I asked you to draw about the red. What is the ratio?
3-1.
Cepat.
3-1.
Jawab. Apa ratio dia?
3-1.
Kenapa I tak dengar ni? I tak buka sound ke? Oh, I tak buka sound. Okay, sorry. Seorang jawab?
3-1, teacher.
Yes. Thank you. Okay, betul. 3-1. Ya, 3-1. Okay. So, patah balik kat slide. Okay. So, 3-1. Okay, 3-1. Kadang-kadang I sebut 3-1. Student ingat 3-1. Salah. I tak nak sebut dah 3-1. So, kita sebut 3-1. Okay, this is monohybrid. Ya, monohybrid ataupun first Mendel law. Okay, so kita tengok yang ni. For second Mendel law ataupun dihybrid. Okay, so you nampak kan, kita buat ratio for phenotype. Kita tengok phenotype dia. So the ratio, kita ambil yang dominant dulu. Both are dominant. So round and yellow. So you can count from the Punnett Square. Okay, round and yellow ada 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 round and yellow. Both are dominant characteristic. Okay, so that's why ditulis dekat sini 9 round and yellow. This is the phenotypic ratio. Phenotypic ya, yang kita nampak. Okay, so kalau you nampak shape dia sangat cantik. Dia boleh bentuk satu triangle ni. Okay, so triangle yang besar tu, that is 9 dominant characteristic. Okay, so dia ada lagi 3. Okay, 3 ni, okay, you cari mana 3 ni. Okay, so it consists of round for the dominant shape, but the color is recessive, green. So there are 3 also. Okay, 3 bukan 3 also yang ini. Okay, there are 3. So 1, 2, 3. Dia dekat tengah-tengah. Sini satu, dekat tepi sini satu, dekat tepi sini satu. Okay, big B, small b. Yang ni dia dua-dua ni small k because dia green color, dia recessive. That's why color dia green ya. So dia ada 3. So ada lagi 3. Another 1, 3 ni, this is for constricted and yellow seed. Yang ini, nampak tak square pula. Triangle yang kecil ni. Nampak triangle yang kecil ni. So this is constricted, dia punya shape dia recessive, but the color is dominant, yellow. So dia constricted and yellow. Okay, so here dia ada 3. And last one, there is only one. Both are recessive characteristic. So the genotype must be small b, small b, small k, small k. So that's why untuk Mendel second law or dihybrid inheritance, the ratio is 9:3:3:1. Okay, so after this, if I ask you about the ratio, dah ada dah dalam kepala you all. Oh, monohybrid 3:1. So kalau dihybrid 9:3:3:1. Siapa yang buat kajian ni? Mendel lah. Dia yang come out dengan result ni. So kita belajar benda yang sama. Tak ada orang lagi yang kalahkan teori dia. So memang betul lah dia adalah fathers of genetic. Okay. So kalau you belajar dekat universiti nanti pun still lagi you akan tahu benda ni tapi you akan belajar in detail. Okay, so this is the conclusion for Mendel second law. So Mendel introduced Mendel second law, also known as law of independent assortment. This is the new word for you. Okay girls, listen please put the word in the bio word. The word genotype, allele, locus, everything you put in your bio word. Ya, I harap ada bersemuka, I akan check semua tu. Okay. So independent assortment, what is the meaning? Okay. So kalau first law, okay, Mendel first law ataupun monohybrid, what is the name of the law? Kita panggil apa? Law of what? Law of? Ingat lagi tak? Law of apa?
Law of segregation.
Yes, thank you. Law of segregation means that we separate the allele to take out in one gamete, right? So but for the Mendel second law, kita panggil the law of independent assortment. What is the meaning of independent assortment? Okay, this is the meaning. During gamete formation, each allele from a pair of allele can combine randomly, okay, with any allele from another pair of alleles. Okay, kalau you tengok gambar tadi, we also take out the gamete. Okay, then what happened to the gamete? Okay, to the alleles, pairing of alleles tu, kita boleh keluarkan dia banyak and then dia boleh combine randomly. That's why kita come out dengan Punnett Square. Jadi dia ada banyak possibilities, okay, because gamete dia banyak. Dia boleh pairing dengan mana-mana pairing yang dia ada. Dia boleh jadi both are dominant, dia boleh jadi heterozygote, dia boleh jadi both recessive. So that's why dia punya ratio lagi banyak, 9:3:3:1, because dia carry two pairs of allele. Okay, dia carry two characteristic. That's why untuk law ni kita panggil dia law of independent assortment. Independent ni dia tak bergantung, maksudnya dia boleh campur dengan mana-mana. Dia adalah secara rawak, ya, independent assortment. Okay, for second law ni. Okay, so this is the summary of Mendel dihybrid. Okay, so new combination of characteristic are produced in the F2 generation, namely constricted yellow seed and round green seed. So ini adalah dia punya combination of the characteristics. So F2 generation dia akan basically akan dapat inilah result dia. Okay, constricted yellow seed and round green seed. Okay, so two characteristic, okay, seed shape and color are combined in F1 generation, but later they separate and react freely in F2 generation. Yang ni because there are a lot of gametes, so dia akan combine randomly. Okay, ini tak payah hafal, dia just bagi satu conclusion, okay, to differentiate between the Mendel second law and Mendel first law. Okay, habis about dihybrid. So I want you to draw the schematic diagram. Soalan tu pernah keluar dalam SPM. You tengok dalam buku pasir, saya tak ingat lah, tapi dah lama, 2018 ke macam tu. Maybe boleh keluar untuk you all. Ingat soalan genetik dia either come out in structure or AC. Kalau dia keluar structure, dia keluar schematic diagram, you need to put the genotype. You need to determine the phenotype based on the gene tu. Okay. Kalau dia tak keluar structure, dia isi tempat kosong kan, kalau structure kan. So you answer the question. Dia tak keluar structure, dia keluar isi. So you need to draw the schematic diagram. Kalau genotype you salah, habis you punya schematic diagram salah. Itu satu dia punya risiko. Tapi kalau betul semuanya, you akan dapat markah dengan senang. Okay, sebab tu basic kena kuat. Kalau I sebut homozygote, you tahu apa. Heterozygote apa. Sebab soalan dia akan bagi tahu macam tu. So tak apa, nanti kita akan tengok beberapa contoh soalan. Okay. So last slide. Okay, about gene and alleles. Okay, yang ni pun I dah discuss dengan you all. Okay, genes and alleles. Alright, so what is the definition of locus? You belajar juga dalam maths kan, locus. Okay, locus is the location, location, specific location of a gene in a chromosome. So each gene must have their position ataupun their location, okay, at the chromosome. Okay, especially homologous chromosome lah. So kalau dia pairing, kita panggil dia alleles. Okay, so each chromosome carries many genes. Okay, many genes. So in human, the number of genes that code for proteins in a set of haploid chromosome is estimated to be 25,000 gene. Banyak tau untuk code kita punya protein. Protein kita ada banyak jenis. Protein tu yang akan determine what type of enzyme, what type of hormone and also the cell. So banyak ni, ada 25,000. Okay. So allele, okay, which represent a gene is located at the same locus as the gene. Okay, so allele dengan gene ni sebenarnya dia sama je. Cuma allele dia ada pairing. So bila dia pairing at the same locus, so dia panggil sebagai a pair of alleles. Dia ada S lah, a pair of alleles. Dia sepasang. Actually dia adalah gene. Okay. So kalau kita tengok gambar ni, you akan nampak dengan jelas. Alright, so this is homologous chromosome. Okay, bila kita jumpa homologous chromosome, ada tanya hari tu, when we can see this structure in the cell division. During what phase? Ingat lagi tak? Ada sebut jawapan. Apa?
Prophase 1.
Prophase 1. Lambat sangat ada nak tersebut jawapan. Okay, prophase 1. Okay, good. So prophase 1 ingat dia pairing. So nanti kalau dalam metaphase 1 dia dah align. Homologous tu dah align. So bila anaphase 1 then baru the homologous chromosome separate to the poles. Okay. So look at this. Okay, so in this homologous chromosome, okay, they carry genes. Okay, so the genes, okay, must be located at the specific locus. Alright. So example for this allele that represent gene. Kenapa dia allele? Because they pairing ni. So they carry the same gene. Example, height, okay, maybe your color of skin. Okay, so kalau you nampak yang ni dominant kan? A, big A. Yang ni recessive ya. Okay, so ciri kita yang ada dalam badan dia akan follow yang dominant. Macam badan you sekarang, kita ada 46 chromosome. So 23 from your mother, 23 from your father. So dia pairing, dia pairing, jadilah 46. Okay, so bila dia pairing, dia akan determine, okay, which one is dominant, which one is recessive. Kita akan follow yang ciri dominant. So kalau rambut you kerinting, sebenarnya you follow yang dominant trait. Tapi tak tahulah, maybe daripada your mother or your father. So you can see at the phenotype for your mother and father. You tengoklah siapa yang rambut kerinting. So that one must be dominant. Okay, so yang ni you nampak, okay, this is also the a pair of alleles, so both are dominant. Okay, so yang ni dia bagi satu contoh. On these two loci, okay, dia ada dua location ni. So the gene are homozygous because of similar allele. Okay, kenapa dia homozygous? Because this one big B, big F. So kita panggil dia homozygous dominant. Kenapa homo? Because dia same letter, both are big. And then kenapa dominant? Because dia big F. Okay, yang ni homozygote recessive. Okay, so kalau homozygous recessive, small j. Kenapa dia homo? Because dia same, small. Kalau one big, one small, dia tak jadi homo dah. Dia jadi apa? Hetero. So that's why dia kata yang ni, on this location, locus, okay, the genes are heterozygous because of different allele. One big and one small. But there are still a pair of alleles. Cuma dia hetero. Satu big, satu small. So bila satu big, satu small, nanti nak determine dia punya next generation, dia mesti follow the dominant. Kalau yang ni dua-dua adalah dominant. So confirm anak dia ataupun orang yang ada kromosom ni akan follow yang dominant. Okay? Okay, habis. Boleh? Okay, so I akan habiskan sampai sini je hari ni, 11.3. So next class, okay, we will continue about 11.4. Dia lebih menarik because we learn about human. Okay, what are the characteristic inherited in human? So dia ada juga setengah tu ada diseases. They carry the diseases lah, inherited diseases, penyakit yang diwarisi. Okay, contoh color blindness, itu adalah penyakit yang diwarisi. Hemophilia, ya. Okay, penyakit genetik. Okay, so do you have any question? Ada soalan? Okay ke? Ada soalan tak sebelum I habiskan kelas?
Tak ada cikgu.
Tak ada, boleh. Okay, so hari ni kelas kita sangat ringkas. Okay, setengah jam je kan. Sebab benda yang cikgu ajar pun senang. Okay, dihybrid. So after this, okay, please draw the schematic diagram. Okay, and then you take picture. Okay, take picture and then send to me. Okay, PM. Ada yang hari tu dia hantar dekat I, bagus. So I boleh tick, I boleh check apa kesilapan you all. Okay, so hari ni bagus semua hadir. Okay, good. Okay, so Izzati nanti tengok mono hybrid inheritance sebab tu Izzati tak datang seorang. Nanti video ada tapi cuma saya belum send lagi. Okay, nanti I akan send dalam masa terdekat. Okay, so I think that's all for today. So please do the tutorial. Okay, draw the schematic diagram and fill in the blanks. Okay, bye-bye.
Thank you, teacher.
Okay, welcome.
Thank you, teacher.
Sama-sama. Okay, bye.